Hi, my name is Portia, and I'm here to present Chapter 17 of Chemistry 104. Chapter 17 talks about nucleic acid. Nucleic acid are made of smaller units called nucleotide. Each nucleotide has three major components. So, a nitrogen-containing base, pentose sugar, a phosphate group. So there are two major categories on the nitrogen bases, and these are the purines, the adenine A and the guanine G. They are a double ring structure, and the pyrimidines, that's the cytosine C, thymine T, and the uracil, U. They have a single ring structure. So DNA contains A, G, C, and T, and the RNA contains the A, G, C, and U. This difference helps distinguish the two nucleic acids and influences how they interact in protein synthesis. So RNA contains so there's sugars in the DNA and the RNA. So RNA contains the ribose, and the DNA contains the deoxyribose. The deoxyribose, and which lacks one oxygen atom on a carbon too. So this small structure differences leads to major difference in stability and function. DNA is more stable because it must store genetic information at the long term. And nucleoside versus nucleotide. It's important to understand that distinction between a nucleoside and a nucleotide. So a nucleoside has a sugar and a nitrogen base, and a nucleotide has a sugar, nitrogen base, and at least one phosphate group. So the primary structure of the nucleic acid. So nucleotides are connected by phosphodiester bonds. These bonds link three hydroxyl group of one sugar to the 5-phosphate group of the next. As a result, the chain has two ends. So the 5-phosphate and the 3-hydroxyl end. So the sequence of these of the bases along a nucleic acid chain is unique to every organism and contains the instructions for making proteins. The DNA double helix. So DNA is famous for its double helix structure. Think of its twisted ladder. So the sugar phosphate backbone forms the sides, and the nitrogen bases form the ranks, and the complementary base pairing occur as a pair of T by 2 hydrogen bonds. Then the gene pairs with C by 3 hydrogen bonds. So this pairing ensures accuracy when DNA is copied and when proteins are made. So the DNA replication. So before a cell divides, it must copy its DNA, and this process is known as the DNA replication. So steps: helicase unwinds and separates the two DNA strands. Each strand serves as a template. So DNA polymerase adds complementary nucleotides. Two identical DNA molecules are produced. So the process is called, so when two identical DNA molecules are produced, this process is called the semi-conservative, because each DNA molecule contains one old and one new strand, ensuring accuracy. DNA fingerprinting. So DNA fingerprinting is a technique used to identify individuals. So enzymes cut DNA into fragments which separates into distinctive patterns. So similar to a barcode. So each person has a unique pattern, making this method useful in forensics, paternity testing, and genetic analysis. So RNA plays several roles in the cell. So unlike DNA, RNA is usually single-stranded and much smaller. And RNA uses ribose as its sugar and uracil instead of thymine. And its main functions include carrying genetic information and helping build proteins. So types of RNA. There are three major types of RNA involved in protein synthesis, and the first one is the messenger RNA, that is the mRNA, carries instructions from DNA to the ribosomes. And the second one is the transfer RNA, that is the tRNA, and brings amino acid to the ribosome during protein synthesis. And three, the ribosomal RNA, which is the rRNA, forms ribosomes where proteins are assembled. Each type plays a unique and essential role in turning genetic information into functional proteins. So genetic code. So the genetic code is based on codons and three base sequences on the mRNA. Each codon specifies one amino acid. For example, AUG codes for methionine, and that the start amino acid. So the tRNA molecules read the codons using anticodons and bring the correct amino acid to the growing protein chain. So protein synthesis occurs in two main stages, and one is transcription. Inside the nucleus, DNA is copied into the mRNA. The mRNA carries the instructions for building proteins, so translation. And the second one is the translation, and that's the second type of protein synthesis, where the protein synthesis occur. So in the translation, in the cytoplasm, the ribosome read the mRNA and the RNA amino acid in the correct order. The sequence of amino acid determine the protein shape and the function. So mutations are changes in the DNA sequence. They can cause the UV radiation, chemicals, X-rays, or viruses. So substitution, one base replaced by another, and the frame shift mutation. A base is added or removed, shifting the reading frame. So mutation can change the protein structure and lead to diseases such as sickle cell anemia or albinism. So genetic diseases occur when mutations affect important proteins or enzymes. For example, PKU results from a missing enzyme needed to process phenylalanine. And albinism occurs when the enzyme needed to produce the melanin is defective. And these diseases highlight the importance of accurate DNA replication and protein synthesis. Viruses are not living cells. They consist of genetic material, either DNA or the RNA, inside a protein coat. They cannot produce on their own and must infect a host cell. Inside the host, viruses take over the cell's machinery to produce more virus particles. So retroviruses such as HIV contain RNA instead of DNA. They use an enzyme called the reverse transcriptase to convert their RNA and DNA. So this viral DNA then inserts into the host genome and produces new virus particles. Retroviruses are particularly dangerous because they can permanently alter the host genetic material. Thank you very much. This is the end of the presentation.